Welcome to G Fun Facts Online. Today, we're not using a time machine to travel to the age of dinosaurs. Instead, we're using something far smaller, yet infinitely more detailed: their fossilized teeth. Imagine holding a single tooth from a Tyrannosaurus rex or a giant sauropod. It's not just a relic, it's a high-fidelity hard drive, an intricate natural archive that recorded the world of the Mesozoic era with astonishing precision. This is the incredible field of forensic paleoclimatology, where scientists are unlocking the secrets of ancient climates, ecosystems, and the very air that dinosaurs breathed, all from the microscopic clues locked within their dental remains. We're going to peel back the layers of these prehistoric time capsules and see what they tell us about the lost world of the dinosaurs. Let's start with why a tooth is so special. Fossilized teeth, and specifically their enamel, are a treasure trove for scientists. Tooth enamel is one of the most resilient biological substances known, capable of surviving for hundreds of millions of years with minimal alteration. This is crucial. Unlike bones, which are porous and can be easily contaminated by minerals from surrounding rock and groundwater over geological time, enamel's dense crystalline structure acts like a vault, protecting its original chemical signature. This makes it the perfect material for geochemical analysis. While the inner part of the tooth, the dentine, is more susceptible to change, it also holds valuable secrets, particularly about the animal's growth. The real breakthrough has been the convergence of paleontology, geochemistry, and advanced imaging technologies, allowing us to read these dental archives with unprecedented precision. So how exactly do we read the chemical language stored in these teeth? The most powerful tool is isotopic analysis. Isotopes are just different versions of a chemical element, with a different number of neutrons. The ratio of these different isotopes in a dinosaur's tooth tells us a huge amount about temperature, diet, and even the air it breathed. Let's talk about oxygen isotopes. The ratio of the heavier oxygen 18 to the lighter oxygen -16 in tooth enamel is directly related to the water the dinosaur drank. That water came from rivers, lakes, and plants, which all got their water from local rain. And here's the key: the oxygen isotope ratio in rainwater is heavily influenced by temperature. So by analyzing the oxygen in a dinosaur's tooth, we can create a thermometer for the Mesozoic. A groundbreaking technique called triple oxygen isotope analysis, which measures all three stable oxygen isotopes, has taken this even further. It allows scientists to reconstruct not just temperature, but the actual concentration of carbon dioxide in the ancient atmosphere. A recent study in the Proceedings of the National Academy of Sciences used this method on dinosaur teeth from the late Jurassic and late Cretaceous. The results were astounding. Around 150 million years ago, in the late Jurassic, CO2 levels were about 4 times higher than pre-industrial levels. In the late Cretaceous, they were about 3 times higher. This confirms a much stronger greenhouse effect and warmer global temperatures. The study also found that global photosynthesis was happening at about twice the rate we see today, likely fueled by all that extra CO2 and warmth. This suggests a hyperproductive biosphere, which makes sense when you consider the energy needed to support the colossal size of so many dinosaurs. Even more, this technique can spot sudden, dramatic environmental events. The teeth of some dinosaurs, including T-Rex, showed unusual isotopic spikes. These are interpreted as evidence of massive, short-term injections of CO2 into the atmosphere, likely from colossal volcanic eruptions like the Deccan Traps in India towards the end of the Cretaceous. So a single tooth can hold the signature of a volcano that erupted millions of years ago, hundreds or thousands of miles away. But what about the dinosaur's own body? Another incredible method, clumped isotope thermometry, lets us measure a dinosaur's body temperature directly. This technique looks at how heavy isotopes of carbon and oxygen tend to clump together in the tooth enamel. The amount of clumping depends on the temperature at which the enamel formed. Studies on Jurassic sauropod teeth using this method revealed body temperatures between 36 and 38 degrees Celsius, very similar to modern mammals. This is powerful evidence in the long-standing debate about whether dinosaurs were warm-blooded or cold-blooded, suggesting they had a sophisticated ability to regulate their own body temperature. Beyond climate, isotopes tell us what was on the menu. Carbon isotope analysis looks at the ratio of carbon-13 to carbon-12. Plants use different photosynthetic pathways, known as C3, C4, and CAM, and this results in different carbon isotope ratios in their tissues. When an herbivore eats those plants, it incorporates that specific carbon signature into its body, including its teeth. By analyzing the carbon in a dinosaur's tooth, we can infer what types of plants it was eating. This is vital for understanding not just a single species, but how entire ecosystems functioned. For instance, analyzing the carbon and strontium isotopes from various herbivores at the same fossil site allows researchers to investigate niche partitioning, how different species manage to coexist by eating different things. A study at the Carnegie Quarry in the Morrison Formation used calcium isotopes to investigate this. The results suggested that different giant herbivores specialized in feeding at different heights, like modern giraffes and gazelles, reducing competition for food. Now let's zoom in from the chemical level to the structural. Much like tree rings, dinosaur teeth have incremental growth lines in their dentine, called von Ebner's lines. It's widely believed these lines represent daily growth. By counting them, paleontologists can determine how long it took for a tooth to form and be replaced, which gives us clues about metabolism and growth rates. This field, known as sclerochronology, also holds the key to understanding ancient seasons. Just as a tree ring is wider in a good year and narrower in a bad one, the spacing and chemistry of von Ebner's lines could reflect seasonal changes. Variations in food availability or water stress from dry or cold seasons might be recorded in the dentine. While it's a developing area, some studies are showing incredible results. For example, cyclical variations in oxygen isotopes have been found along the growth axis of theropod teeth, interpreted as reflecting seasonal changes in rainwater. A theropod tooth from Mongolia revealed a seasonal pattern similar to modern cold, continental climates. In contrast, a tooth from a high-latitude site in Russia showed much weaker seasonal swings, likely because the nearby ocean moderated the climate. It's like finding a detailed weather report from 70 million years ago. Finally, let's look at the tooth's surface. A dinosaur's tooth wasn't pristine. The simple act of eating left a microscopic landscape of scratches and pits. This is the field of dental microwear analysis. The development of a technique called dental microwear texture analysis, or DMTA, has been a game-changer. Using high-resolution 3D imaging, scientists can quantify the complexity and roughness of the tooth surface. Different foods leave different patterns. Tough, fibrous plants create long, parallel scratches. Hard objects like nuts, seeds, or bone leave behind a higher density of pits. By comparing microwear on dinosaur teeth to that of modern animals with known diets, we can make solid inferences. For example, a study on hadrosaurs, the duck-billed dinosaurs, found that their dental microwear became rougher in the late Cretaceous. This suggests a dietary shift to more abrasive foods, possibly linked to the rise of flowering plants, or angiosperms, which often contain abrasive silica particles called phytoliths. Microwear can even reveal behavior. A study on sauropods from the USA, Portugal, and Tanzania found that wear patterns could indicate if a species was migratory. The teeth of camerosaurid sauropods showed remarkably consistent wear patterns across different regions, suggesting these dinosaurs migrated to follow their preferred food sources. In contrast, the more varied wear patterns on the teeth of diplodocids suggest they were more stationary, adapting their diet to whatever was seasonally available. The local environment also leaves its mark. In the semi-arid landscapes of Tanzania, The high levels of abrasive wear on sauropod teeth are thought to be from eating vegetation contaminated with wind-blown sand. So microwear tells us not just what a dinosaur ate, but also about the aridity and dustiness of its world. Let's put all this together with some real-world case studies. The late Jurassic Morrison Formation in western North America is famous for its giants, including the sauropods Camarasaurus and Diplodocus. How did so many huge herbivores coexist? Dental microwear and isotope analysis provided the answer. Camarasaurus, with its more robust teeth, had a diet that included tougher vegetation. The delicate peg-like teeth of Diplodocus were suited for browsing on softer plants. Add in the evidence that Camarasaurus may have been a long-distance migrant while Diplodocus stayed local, and you have a clear picture of niche partitioning, allowing these giants to share the same habitat without starving each other out. Now let's jump to the end of the Cretaceous, to the Hell Creek Formation, the world of Tyrannosaurus rex. Isotopic analysis of T-Rex teeth is what gave us those groundbreaking findings about high CO2 levels and dramatic climate shifts. Microwear on T-Rex teeth gives us insights into its feeding behavior. It was clearly a bone-crushing predator, but microwear can help determine how often it did this, and if its strategy changed with age. Some research even suggests that juvenile tyrannosaurs may have had different diets than adults, perhaps scavenging more often. Finally, let's go to the Kem Kem Beds of Morocco, a massive river system from the late Cretaceous, dominated by a bizarre array of huge predators. Chief among them was the enigmatic Spinosaurus. With its conical teeth and long snout, it was long suspected to be a fish eater. Oxygen isotope analysis confirmed it. The isotopic signature of Spinosaurus teeth is much more similar to semi-aquatic crocodiles and turtles from the same area than to other land-based theropods. This is strong evidence that Spinosaurus spent a significant amount of its time in the water, hunting aquatic prey. This semi-aquatic lifestyle allowed it to avoid direct competition with the other giant predators stalking the riverbanks. Of course, this field isn't without its challenges. The biggest hurdle is something called diagenesis, the alteration of fossils over millions of years. Chemicals from groundwater can seep into a tooth and change its composition, scrambling the original data. This is why careful screening of fossils is so critical, and why the dense, resistant enamel is the gold standard for this research. But the future of forensic paleoclimatology is incredibly bright. Scientists are now integrating multiple lines of evidence from a single tooth, combining isotopic data with microwear and growth line studies to paint a truly holistic picture of a dinosaur's life. New technologies like synchrotron X-ray microscopy are providing even more detailed chemical maps without damaging the fossils. And artificial intelligence is being used to analyze the massive datasets these techniques produce, spotting patterns that might otherwise be missed. The teeth of dinosaurs, once seen as little more than eating tools, are now being recognized as some of the most valuable archives of Earth's deep past. They are a testament to the resilience of nature and the ingenuity of the scientists learning to read their stories. Through the forensic analysis of these remarkable fossils, we are not just reconstructing ancient climates, we are coming face to face with the lost worlds the dinosaurs inhabited. And in doing so, we gain a much deeper appreciation for the long, complex, and dynamic history of our own planet. Thanks for joining us on Fun Facts Online.